Today I want to talk about the kidney. And the kidney is made up of millions of microscopic structures called nephrons. In fact, the nephron is the functional unit of the kidney. That means that that's where all the action occurs. That's where all the filtering of the blood takes place. So let's go ahead and draw a nephron and talk about what actually happens there. Now, when I draw the nephron, I want to go ahead and draw it, label it, and then talk about what each part of the nephron does. It's kind of like an assembly line, and each part is going to be specialized to perform a different function. So let's go ahead and get started. So that's going to be our nephron. The nephron consists essentially of a tubule, a small microscopic tube that's very convoluted, meaning it's very windy or twisty. Now this is going to be a simplified diagram, so you don't see all the convolutions or twists here, but in real life they would be there. Now let's go ahead and draw a blood vessel. So I'm going to draw a blood vessel coming up, going in here, circling around a few times, leaving, dropping down, coming around, and over. So this blood vessel actually comes, it's a branch from the renal artery. And the blood is going to go up in this direction. Eventually it's going to come over here, going to go down this way, up over here, and across to the renal vein, which will eventually exit the kidney. Now let's go ahead and begin to label the different parts of the nephron. So let's go ahead and start here. So this structure right here, all of this is going to be the Bowman's capsule. And the large mass of capillaries right here is going to be the glomerulus. Collectively, Bowman's capsule and the glomerulus are going to be called the renal corpuscle. And that's where all the filtering takes place. Now the next part of the tubule, from here to here, is going to be the proximal convoluted tubule. Now it's called proximal because proximal means near. And near what? It's near the renal corpuscle, where all the filtering takes place. Convoluted means very twisty or windy, so this would be very twisty or windy in real life, but for simplicity we sort of straighten it out. And it's a tubule, a small tube. Now if there's a proximal, there must be a distal, a further tube, and here there is one. So I'm going to go ahead and label that, and we'll call that the distal. And I'll put CT, convoluted tubule. In distal, I like to think of distance or far. So this is farther, at least relative to this tubule here, from the renal corpuscle. And then we have this large loop here called the loop of Henle. And it's going to have two distinct parts to it. It's going to have a descending part and an ascending part or limb. So let's go ahead and label that. That's going to be our descending loop of Henle. And this will be our ascending loop of Henle. And lastly, this is going to be our collecting duct. fact, this collecting duct is going to receive urine from many different nephrons, and eventually this is going to feed in to the center of the kidney and go to the urinary bladder. All right, so those are all our parts. Now that we've drawn it and labeled it, now we can begin to talk about what each part actually does. So let's go over here and start at the beginning where we have filtering take place. In fact, this entire nephron is going to do three major functions. So let me go ahead and write that down. In black, I'm going to write filtering. And then in blue, because I'm going to use a different color for those arrows, I'm going to go ahead and write reabsorption. Now, reabsorption means to take back into blood. When you reabsorb something, it goes back into your blood. And then lastly, the opposite of reabsorption is going to be secretion. And secretion, we're going to get rid of or put into the tubule. All right, put into this tubule at some point in time. So those are our three main functions that are going to be taking place here. Let's go ahead and start with filtration. So we've got blood coming from the renal artery, and there's going to be two renal arteries because there's two kidneys on either side. And eventually that renal artery is going to branch and branch and branch and branch and branch until it's literally microscopic. And that's what this capillary is here. It's microscopic, and because of certain reasons, there's actually going to be a high blood pressure in this capillary network right here, a high blood pressure. And that high blood pressure is going to be very important. There's also going to be small little holes in this capillary, and they have special names. They're called fenestrations. But we're not going to worry too much about that, but there are small holes in there. And those small holes act like a filter, and anything that's smaller than that hole passes through it and gets filtered. And anything that's too large, like a cell or a protein, stays in the blood. It can't get out. So that's what's going to be going on here. So let's write that right here. High blood pressure, and I'll write just an upward arrow. High blood pressure causes plasma, that's the liquid part of the blood, to squeeze or get filtered out, squeeze out of capillary. Okay, so high blood pressure causes plasma to squeeze out of capillary. So that plasma, that liquid part of the blood, and anything dissolved in the blood, like ions and wastes, get put into this Bowman's capsule here, just like that. Let's go ahead and draw these arrows. And we're now going to call this plasma that got squeezed out, we're going to call it filtrate. So I've got lots of filtrate. It's not quite urine yet, but it will be in a little bit. So filtrate. And what does this filtrate actually consist of? Well, the filtrate consists of a couple different things. It's going to consist of a lot of water. It's going to consist of ions, sodium, chlorine, potassium. It's going to consist of amino acids and glucose and other things. But remember, anything that's too big doesn't get passed through these little holes. So for the most part, everything's going to get squeezed out except proteins and except cells. They're just too big. They end up staying behind and staying in this blood vessel. So that's what's going to happen there. So everything goes into the Bowman's capsule. It eventually begins its journey down the tubule. And as you can see here, not only do we have a lot of good things getting filtered out, but we also have some bad things here too, like urea, which is our nitrogenous waste. We don't want urea. That's bad stuff. We're trying to get rid of it. If it builds up, it's going to literally poison our nervous system, and that's not good. Now, a lot of these other things are very good. They are precious resources, and we want to reclaim them. So the rest of this nephron, for the most part, is going to be devoted to reclaiming those precious resources and also getting rid of certain things too, which we'll talk about later. So what goes on in the second part of the nephron here, the proximal convoluted tubule? Well, a couple things are going to happen here. First of all, we're going to reabsorb some things. So I'm going to use a blue arrow to indicate reabsorption. So what are we actually going to reabsorb? Well, we're going to reabsorb some of those things I was talking about. We're going to reabsorb water, we're going to reabsorb ions, we're going to reabsorb amino acids, we're going to reabsorb glucose. All those things are precious resources. Now, we're not going to reabsorb all of these right here. Maybe 70–75% of the water and the ions and the glucose and the amino acids are going to get reabsorbed there. But some of this stuff's going to get reabsorbed later on. Now, something's going to get secreted here. So I'm going to use green to show it coming out of the blood vessel and going into the tubule, because that's what secrete means. We're taking stuff out of the blood vessel and putting it into the tubule, and there are transport proteins that do this. So what is that? Hydrogen ions. Now, hydrogen ions, or protons as they're otherwise called, are the acid part of an acid. So the body tends to be acidic. So in order to get rid of that excess acid so that we can be slightly basic, we need to get rid of the acid part of an acid. We need to get rid of those hydrogen ions. And this is one of the places where that takes place. And remember, anything that goes into this tubule ultimately gets eliminated from the body. Now, the next thing I want to talk about is the ascending loop. All right, we're going to skip the descending loop for just a minute because the ascending loop is going to make it possible for the descending loop to do its job. So let's go ahead and start with the ascending loop. Now, with the ascending loop, we're going to lose salt, sodium chloride. So let's go ahead and write that down, NaCl. And that sodium chloride is not only going to go into the blood vessel here, but it's also going to go into the tissues around here. So maybe I'll draw even a larger arrow. So the tissues and the blood vessel are going to get lots of sodium chloride. In fact, we're going to pump so much sodium chloride out, and most of this is going to be via active transport. Now, some of it occurs through passive transport, but a lot of it's going to occur through active transport. And that's going to take energy. It's going to take ATP. Now, what's going to happen here is we're going to build up a lot of salt in this region, and over here too. Remember, from here to here is not that far away. It looks far on the board, but when we're talking about a microscopic nephron, that space right there is very, very, very minute. So even though we're pumping sodium chloride out here, it's going to be all over this whole area. It's going to be over that whole area. Now, when we pump sodium chloride out, usually what happens here is water would follow by osmosis. But something's going to be happening here. The ascending loop of Henle is going to be impermeable to water. And that's going to be important. And we'll talk a little bit about that later on, why that is the case. So let's go ahead and write that down. Impermeable to water. So water would like to follow the sodium chloride out, but it can't. It can't. And what's going to happen here, let me just give you some perspective here. This is going to be the cortex up here, the cortex of the kidney, the outer part of the kidney. And down here, we're getting deeper and deeper and deeper and deeper into the kidney. This is going to be the medulla, the center of the kidney. And as we move from cortex towards the medulla, we're going to have an increasing salt concentration because of this process taking place here. So I'm going to write high solute concentration. And up here I'm going to write low. Near the cortex is going to be a low solute concentration. So as we get deeper and deeper and deeper into the kidney, closer and closer into the medulla, the solute concentration is going to be higher and higher and higher. And that's going to be very important. So we're taking the salt out here. Now let's go back to the descending limb. And what's going to happen here is that water is going to come out. So let me go ahead and show the water coming out. So water moves out via osmosis. Remember, there's sodium chloride around this entire area. So water wants to go from where there's less solute to where there's more solute, or sodium chloride. So water's moving out. And this is going to be very important for reclaiming a lot of water, because water is a land animal's most important resource. So we need to reclaim that water, because land animals do not have ever-ending access to water on land. Now, another important thing here is the descending loop of Henle is going to be impermeable to salt, to sodium chloride. So they're kind of almost opposites of each other. Now, why not just have the salt and the water both come out together? Well, there's going to be an important reason for that. And that important reason is this blood vessel coming around here. Notice the blood is going this direction, where the filtrate is going in this direction. They're going in opposite directions, or they're going counter to each other. This is an example of the countercurrent exchange mechanism. So let me go ahead and write that down here. Countercurrent exchange. We saw this before in the gills, where we had blood moving in one direction and water moving in the opposite direction. We've also talked about this a little bit in the feet of wolves and foxes and birds, where blood goes one direction, and in our blood is moving in one direction away from the foot, and blood is moving another direction towards the foot. So you got nice warm blood coming down to the foot, and we've got cooler blood from the foot going back to the body. Now what's going to happen here is, because these bloods are going in opposite directions, the heat from the incoming blood is going to go into the heat from the outgoing blood. The blood's going back to the body from the foot. And that's going to save us a lot of heat. Instead of losing that heat to the environment, that heat's going to be gained to the blood going back to the body. That's going to be important for a fox or a wolf living in a cold environment. Well, here something similar is happening. We've got filtrate going in one direction, we've got blood going in the opposite direction, and what this allows us to do, we've got a lot of salt in the blood here, and we've got the filtrate going in this direction. That prevents equilibrium from occurring. If we had the salt moving out and the water moving out with it, we would come to equilibrium very quickly, and we would have a limited ability to reabsorb water. But when we've got things going in opposite directions, that prevents equilibrium from occurring and allows diffusion to take place to a much greater degree. So we're going to reabsorb a lot more water using this process than we would if we did not use this process. So that's going to be extremely important. Countercurrent exchange, what I'm going to write here is prevents equilibrium. In other words, it allows us to concentrate our urine. Concentrate our urine. Now what I mean by concentrating urine is, I mean that it allows us to remove as much water as possible from that urine. We need a little bit of water in the urine to help make the nitrogenous waste, the urea, soluble, but we don't want to lose too much water. And that's what this process is all about. It's about taking back as much water as we can. Now, let's go ahead and move up to the distal convoluted tubule. Now, a couple things are going to happen here. First of all, we're going to go ahead and we're going to reabsorb some calcium. So let's show some calcium coming out of this distal convoluted tubule and going into that blood. So that's reabsorption taking place there. I'm using blue, and that's going to be calcium. And this is going to be under the direction of parathyroid hormone, or PTH. All right, PTH, parathyroid hormone. So the more parathyroid hormone that is released, the more of the calcium is going to get reabsorbed into our blood to help maintain those calcium levels for proper nerve and muscle function. Now, in addition, we're also going to have reabsorption, I guess I'll show that over here, of sodium. And we're going to have secretion of potassium. Now, both of these are going to occur under the direction of mineralocorticoids, especially a mineralocorticoid called aldosterone. So I'm going to write mineralocorticoids here. So mineralocorticoids. These are hormones that are going to promote the reabsorption of sodium and the secretion of potassium. Now, it turns out that sodium is extremely osmotically active. That means if we reabsorb sodium, we're also going to have a tendency to reabsorb some water along with it. And that's going to help promote higher blood pressure, because we don't want our blood pressure to be too low. That's bad too. So this is going to help maintain a proper blood pressure here. Now, another thing that's going to happen is we talked about hydrogen being secreted here, hydrogen ions, and that is also going to happen here, although not under the direction of mineralocorticoids. We're going to secrete hydrogen ions. Again, that helps prevent an acidic condition from building up. Now, the last thing I want to talk about here is the very last part of the distal convoluted tubule and the collecting duct. I'm going to go ahead and I'm going to use a blue marker for that because we're talking about reabsorption. So we're going to reabsorb some additional water here and some additional water from the collecting duct. And I'll write water, H2O, in there. And this additional water, in a sense, we're going to fine-tune our urine here. What's going to happen here is if we're out, let's say we're out on a hot day or we're playing a sport and we're sweating a lot. If we're sweating a lot, we're losing a lot of water in our sweat. We don't want to lose additional water in our urine. So what's going to happen is the body is going to release a hormone called antidiuretic hormone. And I'm going to write ADH, antidiuretic hormone. An antidiuretic hormone is going to promote the reabsorption of water so that we don't urinate out the rest of the water that we have left. Now, if antidiuretic hormone is low, for example, maybe you're in a classroom and you've got a bottle of water and you're drinking this bottle of water and you go through a quart of water in a single period. Well, you've got excess water in your body now. So your body is going to say, well, I'm going to produce very small amounts of antidiuretic hormone. I don't want to produce much antidiuretic hormone. I want to get rid of that excess water. So when I've got low amounts of antidiuretic hormone, I'm not going to reabsorb excess water and it's going to go out. I'm going to urinate that excess water out. If I want to reabsorb lots of water because I'm dehydrated or will be soon, then my body's going to produce lots of antidiuretic hormone. And that's going to promote the reabsorption of water into my blood, and I'm going to retain as much water as possible. And that's going to be it for the nephron.